Hello, everyone. Welcome to the HPL seminar this week. Today, it is my pleasure to introduce our speaker, Dr. Eng Kuan Moo. Eng Kuan got his bachelor's degree in engineering at the University of Malaya, Malaysia in 2009. In the next year, which is 2010, he started his PhD right away at the same university, which I think it is impressive. During his PhD, he worked on the chondrocyte biomechanics using both in situ and in vitro models, and under the supervision of both Drs. Belinda Murphy and Noor Azzuan Abu Osman. And in 2014, Eng Kuan achieved his PhD degree. Then he came to University of Calgary as a postdoc, working with Dr. Walter Herzog to work on tissue and cell mechanics and imaging. Specifically, he has worked on how articular cartilage behaves in response to mechanical loading at different structural levels down to cell level. He also has worked on muscles, such as distribution of sarcomere spacing in a whole muscle, which is somehow related to what he is gonna present today. Most of his work have been recognized really well at multiple conferences, and one of the best accomplishments is that he won the Promising Scientist Award at the International Society of Biomechanics in Calgary, 2019. As one of future plans, he is moving to Finland next year for Marie Curie Fellowship and start working on tissue engineering problems. Aside from his academic career, he likes outdoors and the Rocky Mountains, and he also enjoys hiking, bouldering, road biking, and mountain biking. In this summer, he did road biking to Canmore for the first time. So I assume that Calgary is one of the best places for him to stay. But today, he will be presenting part of his recent work entitled as Sarcomere Length Non-Uniformity in Single Myofibrils and Entire Muscles. All yours, Eng Kuan.
Thank you, Songwan, for nice introductions. And thank you for inviting me and give me this opportunity to present my work today. So we rely on skeletal muscles to power our daily movement. A skeletal muscle can adopt various shapes, such as fan-like, spindle-like, or even circular shape, depending on the function it is associated with. Beneath each unique shape lies the exquisite structure that combines billions of contractile units together for force production. In general, there are 5 structural levels going from the macro scale all the way to the molecular scale. Here, we have the whole muscle as the organ, then to the tissue level in the form of fascicle, and then to cellular level in the form of muscle fiber, then to the subcellular structure called myofibril. I will explain the 5th one in the next slide. And due to the unique structural arrangement, skeletal muscles have very different appearances when you look at them cross-sectionally or longitudinally. From this longitudinal section, it is obvious that the skeletal muscle are striated. The striation patterns contain important structural and functional information of the muscles. Now let's focus on this small area for the next slide. Here we see the muscle stripes in the form of alternating bright pink and dark pink bands. Using a more powerful microscope and zoom in at the yellow box, we can visualize the 5th structural level I mentioned in the previous slide. It is the smallest contractile unit of a muscle, a sarcomere. A sarcomere is defined by the region between 2 Z lines. It contains light isotropic band, or I band, that are made of actin filaments. and dark anisotropic band or A band, which are composed primarily of myosin molecules, but also allowed actin molecules to slide in. Now we have some basic understanding of the muscle structure. How do we relate the structure to the function? And by function, I mean muscle force. The structure-function relationship of skeletal muscle was established from the sliding filament theory of Gordon, Huxley, and Julian in the 60s, in which active muscle force was predicted based on sarcomere length. This is a classical sarcomere force length relationship of a skeletal muscle derived from the sliding filament theory, with the y-axis being the muscle force and the x-axis being the sarcomere length. It describes the maximum muscle force capability of the muscle, which uniquely depends on the amount of overlap between the actin and myosin molecules. The force length curve has an inverted U shape and consists of the ascending limb, plateau, and the descending limb, and can be derived theoretically from the lengths of the contractile proteins, actin, and myosin filaments. And typically, myosin length is constant at 1.6 micrometer across all vertebrates, while actin filament is species dependent and varies between 0.9 to 1.2 micrometer. So at this point, I hope everyone can appreciate the association between muscle morphology and function. We can now move to the research topic of interest, sarcomere length non-uniformity in single myofibrils and entire muscles. This talk will be divided into two parts based on the muscle hierarchical level. At the myofibril level, we will investigate the relationship between sarcomere length non-uniformity and sarcomere length instability. At the whole muscle level, we will look into sarcomere length non-uniformity during isometric steady state and compare the results with those found in the single myofibrils. We'll start by defining what we mean by sarcomere length non-uniformity and instability. Consider that we have two myofibrils, each containing 10 sarcomeres. The sarcomere length is defined between two Z lines. Here, both myofibrils have an average sarcomere length of 2.7 micrometer. In myofibril 1, the sarcomere length distribution is perfectly uniform. In myofibril 2, despite having the same average sarcomere length, the distributions of sarcomere lengths is highly non-uniform, with a 1.6 micrometer difference between the longest and the shortest sarcomeres. With the example, we define that sarcomere length non-uniformity as the deviation of sarcomere lengths from the average value in a group of sarcomeres at an instant of time. For sarcomere length instability, it indicates the rate of change of individual sarcomere lengths over the course of steady state isometric force. Sarcomere length non-uniformity and instability has played a major role in the explanations of some of the most important and fundamental properties of skeletal muscle, such as force creep, residual force enhancement, and residual force depression, which occurs on the descending limb of sarcomere force length curve. Let's take residual force enhancement as an example. Here, we have the sarcomere force length curve presented in solid white line, showing only the plateau and the descending limb. The white dotted line indicates the passive force. Now let's say we have two sarcomeres on the descending limb with an average force indicated by the yellow dotted line. Based on the sarcomere length non-uniformity theory, after active stretch, The sarcomere on the upper part of the descending limb in blue will shorten at the expense of the other sarcomeres in orange as it pop and be caught by the passive force. The end result is enhanced force. According to this theory, sarcomere length will become more non-uniform and unstable upon active stretching. However, previous studies from our group did not find increased sarcomere length non-uniformity nor unstable sarcomeres following active stretch. Despite playing important role in muscle mechanics, there's a lack of understanding of the intricate relationship between the non-uniformity and instability of sarcomere lengths. Therefore, our goal was to measure sarcomere length instability and sarcomere length non-uniformity within the same myofibrils during isometric contractions before and after active stretch. Such measurement also allows us to investigate the relationship between sarcomere length instability and sarcomere length non-uniformity. Our experimental setup is summarized in this simplified figure. Now we have isolated myofibrils lying in a chamber that sits on the microscope stage. And then a glass needle and a cantilever will be positioned via micromanipulator to pick up a myofibril. Under the microscope, we have a view as shown in this inset. On the left is a glass needle, and on the right is a cantilever. The striated specimens in the middle is the myofibril specimen. Jacuzzi method was used to either activate or deactivate the myofibril by adding calcium solution or relaxing solutions to the sample chamber. This graph summarizes the experimental protocol. The y-axis indicates the motor positions expressed in average sarcomere length, and the x-axis shows the time. The passive and active states are indicated in blue and yellow, in red, respectively. By controlling the activation state and myofibril length, we went through 5 steady states. Starting from the left, passive short at 2.5 micrometer, and then pull to 3.2 micrometer for passive long. Calcium activation started for active long state. Under activated condition, the myofibril was shortened to 2.5 micrometer for active short, before being pulled back to 3.2 micrometer for active post-stretch condition. Here, the active shortening between active long and active post-stretch condition allows direct comparison between sarcomere length non-uniformity and sarcomere length instability during isometric contractions before and after active stretch. To add clarity, I will provide you with the mathematical definitions for non-uniformity and instability. Sarcomere length non-uniformity is measured as the coefficient of variation of sarcomere lengths, which is divisions of standard deviation by the mean value. And for sarcomere length instability, we represent it through absolute slope of best fitting linear lines to individual sarcomere length time curves. I will clarify this with an example in the following slide. This graph shows the sarcomere length changes with time in a myofibril containing 12 sarcomeres, with the y-axis showing the sarcomere length and x-axis the time in seconds. The red curve represents the mean sarcomere length in various steady state, starting from the left, passive short, passive long, then active long, active short, and active post-stretch. I will resolve this curve into individual sarcomere length changes. Calculations of sarcomere length non-uniformity can be done by taking the standard deviations of sarcomere lengths at a time point and then divide it by the corresponding mean value. To measure sarcomere length instability, we isolated time segments for each steady state and then fitted linear lines to every curve. The absolute value of the resulting slope of the fitted line is taken as a measure of sarcomere length instability. Here's the result. The graph on the left shows sarcomere length non-uniformity in terms of coefficient of variation, and the graph on the right indicates sarcomere length instability in nanometer per second. Under passive condition, sarcomere length non-uniformity and instability reads about 8% and 10 nanometer per second respectively. Upon calcium activation, sarcomere length non-uniformity increased by 2.5 to 3 times, but these values are similar in isometric contraction before and after active stretch. For sarcomere length instability, isometric contraction led to 6 times growth to about 70 nanometer per second. But this value dropped significantly to about 40 nanometer per second following active stretch. So in this slide, we organized the data a little bit differently. On the y-axis, we summed the instability values of individual sarcomeres and normalized the resulting value by the number of sarcomeres present within a myofibril. On the x-axis, we used sarcomere length non-uniformity developed at the start of a steady state. With this data, we try to address the question, does sarcomere length non-uniformity always correlate with sarcomere length instability? When the myofibrils were active long or active short, sarcomere length non-uniformity showed positive correlation with sarcomere length instability. But following active stretch, this positive relationship disappeared. To summarize, our results show that sarcomere length are equally non-uniform before and after active stretch. We provide evidence that sarcomere lengths become more stable in isometric contractions following active stretch. Previous studies by Edman et al. And Telly et al. Also found similar observation. Now, this increased stability may be explained by the engagement of structural braces such as titin, which is recognized nowadays as the third filament in a sarcomere. We also show that sarcomere contraction dynamics were altered following active muscle stretch, such that the instability was no longer correlated with sarcomere length non-uniformity. Importantly, we show that these two parameters describe different phenomena and should be considered separately. Now that concludes the first part of my talk, and we'll now switch gear to move to sarcomere behavior at the whole muscle level. Unlike myofibrils, where only a few proteins such as titin or nebulin that could provide structural support, there are abundant of collagenous proteins in the extracellular matrix of intact muscles that offer structural reinforcement to the sarcomeres. This is especially true on the descending limb of the force length curve, where passive forces from the matrix and the intracellular titin grow exponentially. Under such mechanical environment, how would sarcomeres behave? And specifically, we wanted to know if sarcomere length non-uniformity is present in intact muscle. And if so, how does it compare with the sarcomere length non-uniformity observed in isolated myofibrils? Also, if sarcomere lengths are non-uniform, would this non-uniformity remain constant during the isometric steady state? To answer this question, we need to visualize sarcomeres in intact muscle. To do that, we used multiphoton laser scanning microscopy to perform second harmonic generation imaging of sarcomeres in the intact muscle. Intact muscle tendon unit of mouse tibialis anterior was connected to a motorized micromanipulator. The muscle tendon unit was held first at reference length and then stretched to 5 target lengths, during which sarcomeres were imaged. The two curves here shows the force traces for the relaxed and the activated trials. At the target lengths, which is indicated by the white arrow, the tibialis anterior was either electrically stimulated or not for the activated and relaxed trials respectively. Sarcomere images were taken during the 400 milliseconds quasi-steady state, highlighted by the gray boxes here. Representative images of the sarcomeres are shown hereby. After that, we plotted the data on sarcomere force length curve for mouse TA and grouped the data based on their locations on the force length curve. Group 1 is on the ascending limb, while group 4 sits on the lower part of the descending limb. Then we did data analysis similarly as described earlier for myofibril preparation. Here's the results of sarcomere length non-uniformity in intact muscle tendon unit. The y-axis indicates the coefficient of variations in percentage, and x-axis shows the different groups from group 1 to group 4. In the passive state, sarcomere length non-uniformity ranges from 4 to 6%. Upon activation, the sarcomere length non-uniformity increased to as high as 10% in group 1. But as we move down the descending limb from group 2 to group 4, the non-uniformity decreased to the point where sarcomere length non-uniformity was no longer different between activated and relaxed trials, as shown in group 4. So how do these values compare with those found in myofibril? We recorded the passive long state stretch myofibril to an average sarcomere length of 3.2 micrometer. So that corresponds to group 4. Here's the value of passive long state in myofibril. It reads about 8%, which is about 2 times higher than the sarcomeres in intact muscle. What about active long state? It is beyond the boundary of this graph, so we have to readjust the y-axis. It turns out that the sarcomere length non-uniformity in myofibril is more than 6 times higher than that in intact muscle tendon unit. This shows that the extracellular matrix functions to regulate the degree of non-uniformity in the sarcomere lengths. We were also curious on the development of sarcomere length non-uniformity during the quasi-steady state of isometric contractions. So we plot the data out on these 2 graphs. On the left shows the data from the relaxed trials, while the data of the activated trials are shown on the right. As usual, coefficient of variation is on the y-axis and time on the x-axis. Let's take a look at the data in the relaxed trials first. During the 400 millisecond quasi-steady state, sarcomere length non-uniformity remains relatively constant. And next, we plot the data from activated trials. This data suggested that the sarcomere length non-uniformity decreases with time for all groups at a rate of about 2 to 3% per second. In other words, sarcomeres in intact muscle constantly rearrange themselves to correct for the sarcomere length non-uniformity developed in the beginning of activation. How does this behavior compare to those in myofibril? Here's a graph showing the rate of change of non-uniformity. For group 4, it is minus 2% per second. In the active long state in the myofibrils, it shows the reverse trend as the non-uniformity grew at a rate of about 1% per second. So why sarcomere lengths in intact muscle tendon unit became more uniform during isometric contractions? Since the phenomenon occurs primarily in the activated trials, the force production mechanism may be involved. Previous studies by Linari et al. And Fusi et al. Found that the force-mediated activation of myosin filaments may help stabilize the non-uniform sarcomere lengths during isometric contractions by awakening portions of the so-called super-relaxed cross bridges. Shapatra-Tilp then suggested that a threshold value may exist for sarcomere length non-uniformity, after which force-mediated regulation by the thick filament becomes ineffective. So in our case, the high non-uniformity value in myofibril, which is about 25 to 30%, may have passed this threshold, thus leading to increasing sarcomere length non-uniformity with time. However, the mechanism above cannot explain the decrease in sarcomere length non-uniformity with time as observed by us in the intact muscle tendon unit. So that concludes the presentation of the second part. Overall, I showed that the sarcomere length non-uniformity and sarcomere length instability are two different phenomena. Although they are involved in the development of residual force enhancement, they could not exclusively explain its occurrence. Further studies should be conducted in unraveling the molecular mechanism associating non-uniformity and instability of sarcomere lengths. And finally, we show that the extracellular matrix of skeletal muscle functions to keep sarcomere length non-uniformity in check. But very little is known about why this non-uniformity decreases with time during isometric contraction. Hopefully, theoretical models could help us understand this phenomenon in the near future. Before I end, I would like to thank my supervisor, collaborators, and funding agencies to make this research possible. Thank you very much for your time, and I'm open for any question from you. Thank you.
Thank you, Eng Kuan, for the great talk. I really enjoyed it. If you have any questions to our speaker, please use raise hand button at the bottom of your screen. Then I'll call on you when it's your turn. I'll open the seminar to the questions. Does anybody have any questions for Eng Kuan? Actually, I have a question for you, Eng Kuan.
Yes.
So I might have missed in your talk, but I guess you measure the sarcomere non-uniformity at one spot on the intake full muscles on the rats. So I'm just curious that would the location of the where you measured it relative to the entire muscle length also kind of affects your results, like measuring it at the middle of the muscle or the end of the muscles, like close to the tendon? Do you think the stretching magnitude would be a bit different, and then the sarcomere non-uniformity could be also different?
Yes, that's a good question. We have done some measurements at different locations across the tibialis anterior of mouse. And indeed, the sarcomere length distributions are quite different for especially at the proximal end of the muscle, but not between the mid belly and the distal end of the muscle. So in this particular talk, I'm presenting the result that was collected at the mid belly of the muscle. Yeah. And so, yes, we acknowledge that there would be differences. There could be differences if this sarcomere length is imaged at the different locations of the muscle. Yeah.
Okay, thank you. I see Art, you are raising your hands. Please go ahead.
Yeah, I'm just curious what you mean. You mentioned that the thick filament helps stabilize the sarcomeres, and I'm wondering, what are you referring to? Is that just the ascending limb of the force length curve, or is it something else?
Yeah, it's something else. It's like it's kind of new in this field. So Linari et al. First published this one in Nature paper, and then they found that in the regular calcium-related activation, and typically involve low forces, only about 20 to 25% of the cross bridges on the myosin filament. And then if you have high forces, the force that is transferred to the existing cross bridges will activate or awaken those that are in the super relaxed states. So it increases the number of cross bridges at the end of the day, and thereby like increasing the stiffness or the force generation.
So the idea is that some cross bridges, when they're under high tension, they will facilitate activation of neighboring cross bridges.
Yes, yes, that's what that was reported by Linari et al. And Thusi et al.
And then how do you know that that's due to the thick filament? Because couldn't it also be something about local effects on actin or tropomyosin or something?
I do not know. There was the study done by them, but what they did is that they measured the repeating length. They did it by small angle diffraction methods, which I'm not familiar with. But somehow they measure that there is a changes in the repeating length between myosin head. So typically when the myosin is in the super relaxed condition, they will have this helical tract around the myosin filament. And when it's awakened, that head will just deviate away from the main axis of the myosin filament, so that it will be closer to the actin filaments, like kind of like opening up. Yeah, so that's what they measured on the thick filament conformational changes, and then they address they attribute that to the thick filament rather than caused by the calcium-mediated activation.
I see. Great. Thank you.
No problem.
Brent, please go ahead.
Thanks, Eng Kuan. Have you guys done the experiment where you quantify non-uniformity and you passively stretch it and you quantify it or you activate it or whatever, and then you like relax it, and then you do it again. Like, I guess what I'm trying to get at is would you always see the same non-uniformities if you were able to do this experiment multiple times, meaning that it's always the same sarcomere that's popping or whatever? If you
That's excellent question. I think Walter will be very happy that you asked this question, because right now I think Meng Lee, Walter's new, relatively new master student, is going to address this problem. So what she's gonna do, she's gonna activate and deactivate the muscles for a couple of times in the myofibril, and follow the changes of the sarcomere length, individual sarcomere length, during that repeated activation deactivation cycle, and that can answer the questions. That was also the question that that intrigued us.
Yeah, because that's super important for maybe what's determining the non-uniformities right now. So let's say that it's always the same sarcomeres that are, I don't know, weaker or of lower quality or whatever. Mm-hmm. Like, so what are some current theories that people have right now? Like, why theoretically would one sarcomere be weaker than another one?
Why would it be like that? I have no idea.
Come on.
Based on what I read from Walter's material, he would suggest that such non-uniformity can increase the flexibility of the muscle, so increase the range of motion.
Okay, but you've got the same amount of force that's going through this whole thing. Like, so nobody ever thinks that there's the differences in thin filament densities, or there's something different about the number of myosin heads, or I don't know.
Yeah, that is what people have been thinking, like, oh, the contractile proteins, the amount of contractile proteins within a single sarcomere differs between adjacent sarcomeres.
Okay, great, thanks.
But nobody has actually looked into it, as far as I understand.
Okay, all right, cool, thanks.
Can I jump in here, Song-Wan?
Yeah, surely, please.
Yeah, because because Brent, I'm thanking Brent forever and ever and ever, because I think he has asked exactly the questions that I posed to Meng Lee, who's exactly looking at that. And I think just for the rest of the people that are there, it might sound really strange, but the question that you asked, nobody actually has an answer. So when you repeatedly activate or deactivate, activate, deactivate, we don't really know whether or not this is a completely random effect and just happens to be different every time, or whether or not it's determined somehow by structural or contractile proteins, or the number of proteins, the number of actins and myosins that you have in a given sarcomere. And so it's a very basic question, and nobody has the answer. And I hope that Meng Lee is among the audience here today and listens that actually somebody had the same question that I had. And then the answer that Eng Kuan also gave, and again, it's a very fascinating one, is that nobody has ever measured in a myofibril how many myosins and actins you have from one myofibril to the next, to the next, to the next. I think people just assume it's about the same, but when you look at the myofibril, they're, under the microscope, some sarcomeres seem to be distinctly thicker than others. And I hope Ruth Seraten is on the call here as well, because we started once exactly doing those measurements in sarcomeres, the number of myosins in a single sarcomere. And we developed a technique that we actually can do that, but then we wanted to do it in a repeat sarcomere. It was a summer student, but then the summer was done and we never did it. But it's something that I would love to do. I would love to write a paper with a myofibril and having 20 sarcomeres in series and say, okay, the first sarcomere has 600 myosin, the second one has 620, the third one has 380, that type of very basic information, actually nobody has ever looked at that. So thank you for asking that fabulous question.
Okay, I see Eric, you have a question for Eng Kuan?
Yes, Eng Kuan, very nice presentation today.
Thank
I
you.
was wondering if the amount of non-uniformity is also a function of the activation of the muscle, both at a macro level and at a micro level.
That's a good question. I don't know the answer to that question, because I have not seen anyone doing the submaximal activations of the muscle and see the activation level. So see the sarcomere length non-uniformity as a function of activation level. That could be a future study. Thanks for giving that suggestion. It's a good question, because most of the time you assume that the muscle doesn't go to the full even in a volumetric maximum volume MVC, maximum voluntary contraction. You wouldn't expect the muscle to go as far as the electrically induced maximal activation. So yeah, maybe I would speculate that the non-uniformity developed would be lower compared to what I reported here for the activated condition. Yeah, but there will be an interesting thing to look into.
So one, maybe if I can just quickly jump in again, there is an interesting state when you do submaximal activation for myofibrils, and it's called spontaneous oscillations. And nobody really knows exactly what happens there, but when you do some submaximal activation at a certain calcium concentration, then in your myofibrils, your sarcomeres will actually at a fairly rapid rate go back and forth and get longer and shorter and oscillate relatively quickly. And nobody's really exactly sure why that happens, but there is some interesting phenomena that happen in submaximal activation of myofibrils.
Okay, thank you. I think I saw Rob, you have a question?
Yeah, yes, I do.
Hey, Rob.
Eng Kuan, hi, Eng Kuan. If you exercise a muscle at longer length, you can lay down new sarcomeres at the end of the muscle fiber. And these new sarcomeres, as I understand it, tend to have embryonic type myosin in them. So now you would have a muscle fiber with two types of myosin. The sarcomeres at the end are embryonic type. The sarcomeres in the middle of the muscle fiber are mature type. So if it was a difference in some intrinsic property of the myosin, or whether that includes strength, it might be a perfect opportunity to see a difference in non-uniformity. And I'm sure that's been looked at. Can you comment on what's known about that?
To be honest, I have no idea about this area of research, that you have a new sarcomeres added at the end of muscle fibers, and so I unfortunately cannot comment on that. But that I would, as what you expect, I would presume that the intrinsic material properties between these two types of sarcomeres would be different, or myosin to be different, and therefore causing a different, like a mismatch of material properties, probably leading to a more severe non-uniformity development. I actually don't know, but there will be an area that I can look into in the future. Thanks for the suggestion.
Okay. So sorry, Eric, did you say you have a question?
I do have another question.
Yeah, please go ahead.
So you looked at one type of muscle, which is the TA, which I think has a very unique architecture. At least in humans it does. It has like this very tendinous structure almost all the way throughout the muscle. And so how does the macro architecture, do you think, affect the amount of non-uniformity in the muscle?
The macro structure. So you mean the architecture of the muscle fiber arrangement?
Muscle fiber arrangement, and also how like all these other passive structures also surrounding the muscle. So like, would there be a different type of non-uniformity, say in a bicep muscle that has maybe less tendinous structure around it versus in, say, the TA that you looked at?
I would say I cannot give you a definite answer because I have not looked into it. And I know that in Stanford, Dr. Delpt has looked into this kind of problem in human muscle, but they were using a slightly different approach where they insert a needle containing the optics, and they measured higher value of non-uniformity in sarcomere lengths. But we don't know if that was due to the insertions of the needles or because of this intrinsic highly non-uniform behavior of the sarcomere lengths. So to answer that question, we need a similar type of approach that I'm using here, which is like it's invasive, but it's non-contact because there's no features that is in the way of the muscle during the active contraction. But I would presume the muscle with less collagenous structures to be more non-uniform during activation. That will be my speculation. Yeah.
Thank you. I have more time for a few more questions for Eng Kuan. Since actually I have a more question for you. So in one of your first study, the first study that you showed in the myofibril level, so you compared non-uniformity and sarcomere length instability, and then saying that there's no correlation between the two. It's therefore the two variables need to be recognized as separated ones. But what if you had found actually there are correlated relationship between the two values, and what would that mean? Because I was just curious why the two different variables would be somehow correlated in the initially. So I'm just curious.
So based on previous researchers, for example, Morgan is the most obvious example. In his non-uniformity, sarcomere length non-uniformity theory, he was denoting, like, attribute the unstable sarcomere length, sarcomeres on the descending limb of the force length curve. So because of that unstable sarcomeres, the sarcomeres become more non-uniform during the force enhanced states, and these two somehow is taken as the causal relationship between one another. And we are showing here that yes, the sarcomere length non-uniformity and the instability are correlated in the active long and passive long state, which means that if you don't go to force enhanced state, one can cause the other. And it shows a pretty strong positive correlation. But when you go to a force enhanced state, somehow there's another components that comes into play and changes the behavior completely, and causing this two phenomenon, like non-uniformity and instability, to not correlate anymore. So that's the main message that I would like to send out, is that depending on the scenario, you have to treat sarcomere length non-uniformity and sarcomere length instability differently. Yeah.
Thank you. Walter, please go ahead.
Yeah, maybe Eng Kuan, just to make another comment to that, kind of what you said there at the end. I think if I saw it correctly, and if I remember it correctly, in the myofibrillar data, when you have relative stability or greater stability, I think the values of the slope become relatively small, and I think you have a relatively small distribution, and maybe part of the reason why the correlation between instability and non-uniformity falls away, because you have relatively great non-uniformity, but virtually no instability anymore, and they all hover around zero, and so I think that might be part of it. I'm not sure if that's correct, but I think that might be the case. But the question that I wanted to ask is, you know, I thought it was really interesting, and that at the short lengths, your active and your passive non-uniformity in the muscle, when you do the entire TA, is much bigger. And then when you go to longer lengths, the active and the passive non-uniformity decrease. And not only that, the active at the longest lengths then becomes the same as the passive. How do you kind of reconcile that observation in your own mind?
So the two main differences between the contractions of muscle, the main differences is the length. So by lengthening the muscle, you engage structural components. So I would say that it is the structural components that comes into play in the long muscle tendon unit length that somehow stabilize the sarcomeres. And that structural support does not exist or is yet to come into place due to the slackness of the muscle at that particular length. Say, for example, in the group 1 of the results. Mm-hmm. And therefore, without that support, the sarcomeres become more flexible, but more non-uniform at the same time.
Yeah. So the idea being you have short sarcomere lengths, there's no passive force or not much passive force and no structural element really constraining. So then you get lots of non-uniformity because somehow the activation process, the active process, causes some non-uniformities, and then when you go to very long lengths, at the very longest lengths there's 3.4 microns or so.
Yeah.
Your passive force is presumably as much as your active force, or maybe even more.
It's higher, it's slightly higher.
It's higher, yeah, okay. Yeah. So then your passive force starts dominating. And you essentially see the effect of the passive force, and okay, that makes a lot of sense.
So it's like 60% at the longest length is 60% of passive force, and about 45%, 45% of the active force.
Yeah, yeah. The other, did Rob want to jump in there? Rob, did you want to jump in there?
Not on that point. I've got a different point.
Okay. Why don't you go? And I have a couple more questions at the end. Why don't you go now?
Eng Kuan, I was thinking about different titans. With the diaphragm, it has the high molecular weight titan. And the argument is that that permits a sarcomere to operate out at very long length. And then you get other titans that are much lighter, and the argument is that they operate at lower lengths. If you take muscles of each type with different titans, which have obviously different passive properties, does that change your argument at all? Can you see more non-uniformities in the sarcomeres with a high molecular weight tighten than you see non-uniformities in a low molecular weight tighten?
That is a very good question. I'm not sure I have a good answer for you, but I will expect that there will be difference depending on the tightened isoform within the muscle type. So
Well, in a way, it tests the hypothesis that the passive properties within the sarcomere are contributing in some way to this. You get to, as opposed to it's purely a myosin issue.
Yes, true. I will look into that. I think maybe Venus can comment on that too, because I know Venus has done some studies on tightening with different isoform. Yeah, with a different stiffness, I would expect it would make an influence on both the instability and non-uniformity of the sarcomere lengths. Yeah, good question.
Song one, I can make a quick comment as well here. There are muscles from invertebrates that have not really a tighten, but kind of like a tighten analog. So, for example, in insect flight muscle, there's a tighten analog protein called projectin, and the projectin is somewhere between 500 to 1,000 kilodaltons compared to 3,000, 4,000 kilodaltons. It's much shorter and therefore also much stiffer. And indirect flight muscle of insects have fantastically uniform sarcomeres. I've never really tested them in trying to tear them apart or that type of thing, but I do know they are virtually uniform as far as light microscopy can detect. So I think you are right. I think that happens when the structural elements within the sarcomere become smaller and shorter and tighter, then you'll get exactly that event.
I see Brent. Do you have any questions?
This is a genuine question. I don't know how to word it, so I don't want it to sound the wrong way. But if you zoom in on a myofibril and you observe some kind of structure-function relationship, and then you zoom back out and you start looking at the whole muscle level with all the extracellular matrix and everything, and you don't observe that phenomenon, does that mean that the observations that you're making at the sarcomere level are not important? Does it mean that they're not functional observations, that in general the body really doesn't care about these non-uniformities and things like that? That's just a general question. Like, it has a really, like, should we be looking at more than one sarcomere if you want to look at functional relationships of muscle?
I do believe that you should, because this is like a differences in theory, right? Depending on how you think about it, because a lot of the current paradigm is taking a single sarcomere and upscaling it to a whole muscle level, and then everything derived from that particular single sarcomere model. So what we are observing here is that even though given that the sarcomere should share the same force when they are arranged in series, there is this non-uniformity phenomenon that arises that doesn't make sense. So what is the function of that? And we need to understand it a little bit more. So hopefully we can improve how we think of muscle, how we think that function as a functional properties, or maybe a little bit more far-fetched for the disease. For a certain disease, you have more or less non-uniformity. This is not looked into. I don't think so. So I would say yes, maybe it doesn't matter if you look at macro scale and all you care is just take the end of two muscles and just stretch it and see the force length relationship, and then that's what you care. But at the end of the day, we need to understand these biological tissues from the molecular level so that we can address a different type of questions and hopefully improve our understanding of the muscle in the macro scale. Yeah, I hope that can answer some of part of your question.
I think it's a good answer. I don't think the question has an actual answer. I mean, I was thinking about it more just like not just for muscle, just in the context of bone too, you know? And some people are looking at, you know, nanoscale properties of cortical bone, and you observe phenomena at that level that you don't observe at the microstructural level. And so then the question for me is always, it's more so, does it matter?
Yeah. I think that the one important element for us as researchers is that we should find a connection point between observations at different length scales. And if we could do that, there will be significant advances to understanding of a certain biological material.
I like that. Okay, thanks.
Thank you.
Okay, Venus and then Walter. Venus, please go ahead. If you are around, yes.
Yes, actually, it's a follow-up about the question about the effect of titan structure and titan isoforms. Actually, we have not done experiments in myofibrils on different titan isoforms, but what we have done was that we deleted, or we cleaved titan in myofibrils using trypsin, and we looked at the effect of this on sarcomere length non-uniformities in both active and passive states. Surprisingly, there was no difference. There was an increase in non-uniformity when titan was, I must say, partially depleted, but there was not a significant difference in non-uniformity between when titan was intact and when titan was partially depleted. However, the big difference was when we stretched the myofibril. So we activated, let's say, when titan was intact and when titan was partially depleted, there was no huge difference statistically no difference in non-uniformities. But then when we stretched the myofibrils, the ones with titan partially depleted, they showed a huge increase in non-uniformities. So like this, we must say that so titan was not completely depleted, so we cannot draw final conclusions, but it seems that the big role that is played by titan is when the myofibril or when the muscle is stretched, more than when it is at an isometric, during an isometric contraction.
Yeah, thanks, Venus.
Thank you. Yep, Walter, please go ahead.
Yeah, just also making a follow-up comment about the, you know, sarcomere length non-uniformity, and on that micro level to work on there, you know, how relevant is it or how not relevant. And at the end of the day, you know, it's again an interesting question that Brent raises there. At the end of the day, maybe it's really not that relevant if you look at one or more sarcomeres. Why at the moment, or in the last probably about 70 years, it has been a very important problem, is because people have argued without really great evidence, but people have argued very adamantly that sarcomere length non-uniformity is responsible for a variety of very basic properties of muscle, you know, and Eng Kuan mentioned 2 or 3 of them: creep, force enhancement, force depression. And even today, there is I would argue probably the majority of muscle physiologists would explain, without thinking, force enhancement based on sarcomere length non-uniformity. One of my somewhat cynical comments has been in the past, and I have stopped making it, but in a big muscle, we have billions of sarcomeres. And they always ask, what kind of a brain would you need for billions of sarcomeres to be at the same length, at a uniform length all the time? And from a control point of view, it's just not possible, because you stimulate muscle at a certain, neuromuscular junction, and it takes multiple, tens of milliseconds before that pulse reaches the end of a long fiber. And so in the middle, or where the neuromuscular junction is, your sarcomeres are already contracting, and at the end, they're still passive. And it takes a fair amount of time to get there. And so you'll have these non-uniformities, except if you're an insect indirect flight muscle, where you just have all these tight structural elements, and it's always uniform, but then you can only elongate and shorten by 5%. So you lose the ability to elongate and shorten 100% or 120% like we can in a normal muscle. So the whole idea that in a muscle where you have an enormous amount of excursion, and you can go, from 1.5 microns to 3.5 microns, in order to keep that relatively uniform, would just really be an impossibility, I think. But that's just my own personal opinion there. But I have a couple more questions, Eng Kuan. Mm-hmm. I'm still fascinated, and I had kind of forgotten about it, but I'm still fascinated by the fact that in your myofibrils, the instability is such that sarcomere length non-uniformity seems to slightly increase over time post-stretch, whereas in your whole muscle, during the activation there, the non-uniformity seems to, tends to decrease over time. And I wonder if you have thought about it and have a, kind of an explanation for yourself why that might be.
I do not have an explanation. So I was trying to look into the literatures and trying to find any properties that I'm not aware of, but I cannot find any supports. So the best I could is I already showed it in the presentations, but that could not explain why the sarcomere length non-uniformity decreases with time. It makes sense that the sarcomere length non-uniformity increases with time, because then you have an unstable non-uniform and unstable sarcomeres or myofibril, and then over time you just expect it to aggravate, but it doesn't make sense when you go to intact muscle that trend go to the reverse direction. I don't know.
And then the follow-up question to that, in the myofibril you measure for a long time, but in the muscle you only measure for the 400 or 600 milliseconds. And I know it's not really easily possible to go many seconds in the intact TA, but if you could, do you think the non-uniformity or the change sarcomeres would actually reach a point where they then stay, and they stay quiet and they have no change in length anymore?
That's what I would expect. I would expect that they would decrease, and at a point they would just go to a steady state. Mm-hmm. I don't think it would go lower than what we observe in the passive condition, which is about 2 to 3% of the difference of variations.
And the last comment that I wanted to make, you mentioned Gudrun Schapacher's model about imposing sarcomere stability by waking up, the super relaxed cross bridges, making their rate constant of attachment much faster, and therefore having more cross bridges attached when they wake up, and therefore making that sarcomere stronger. And I was thinking about, that at one point you said the model fails and cannot correct anymore. But, in reality, I think the muscle can always react. And so I was wondering what might potentially be wrong with that model, because I know what you mean. At one point, the force difference is too big and it cannot correct anymore. And so I wonder if the correction factor is not big enough, or if the non-uniformity that she's reaching are too big, or anyway.
Yeah.
More of a comment than something to think about. I'm not sure if you have an answer or want to say something about it.
Yes, I would say, yeah, when the sarcomeres become too long compared to the next, then that force differences become too big to correct for it, and then it's not preventable for it to get worse for the non-uniformity. So in that case, then I was just read that particular paper, but I was not fully aware of the details of that model. So if she also considered the tightening effect, then if she doesn't include the tightening effect, then probably tighten is the role that can help stabilize the sarcomeres.
Yeah, and in that model, I don't think tighten is in there. It's really just an increase in the rate constant of attachment of the super relaxed cross bridges once a certain threshold is reached, and maybe that threshold should be set lower, and maybe the rate constant should be made faster, so that you to react quicker and earlier on before the non-uniformity becomes too big.
Yeah.
I have to talk with her about it.
I was actually emailing her about this the work that we have done on the intact muscle-tendon unit, and she hasn't replied me yet with a new insight. Yeah.
Good.
Hopefully one day she will come back to us with some new information.
See, Art and then Ralph for their last questions. Please go ahead, Art, first.
I was listening to your responses to the questions that Walter just asked, and one of them you answered. I think this was about what happened. Why is it that you have less non-uniformity in the intact muscle? And you said, we don't know. And then I was surprised you said that, because I feel like the bottom of your take-home message says, when embedded in a complex network of reinforcing extracellular matrix, there's less non-uniformity. So I wonder, why didn't you speculate that it could be because of the extracellular matrix?
Yeah, because what I refer to is this particular results. These particular results on the activated states. So when you activate the muscle and leave it there at the isometric steady state, the sarcomere length non-uniformity decreases with time at about 2 to 3% per second rate. So that I don't know why it's the case. I don't think I can use the extracellular metrics as an explanation, because they are passive and they don't shorten over time, and they cannot drive the shortening or rearrangement of the distributions of sarcomere lengths. So
but aren't the cross bridges cycling? So that means that when a cross bridge lets go, there's a short opportunity for the extracellular matrix to do its elastic bit. And then when the cross bridge reattaches, it might have moved a little bit more towards uniform. And then it would just be a matter of So the time part is because of the cycling, and then the direction is possibly due to the extracellular matrix.
Yes, that could be one way to think about it. But because it involves so many muscle fibers within one intact muscle, so it's hard for me to just speculate that is a functional role. But the differences between those two cases is one with intact extracellular matrix, the other one is with almost nothing but the titin and nebulin. Yeah, so I would say that's why I will make a comment at the end of the talk. I hope that the theoretical modelers can do something about this, and then make a big model incorporating a lot of different sarcomeres and muscle fibers with the considerations of the extracellular matrix, and see if that cross-bridge cycling will lead to this rearrangement of the sarcomere length to become a more uniform distribution. Yeah, that will be important for us.
And then I have a basic question related to this, which is what's the relationship between the myofibrils and their neighbors or the extracellular matrix? Do myofibrils slide past each other, or is it more like they are more or less kind of bound together with a stretchy material? Can you tell us whether it's one way or the other, or what do you know about that?
They are connected to each other in a parallel connections by, I don't know what kind of proteins is that. So someone can help me with that, because when you look at the
Desmin.
Sorry, yes, Desmin. Thank you, Walter. When I looked into when you see this picture, yeah, so these images shows contain at least 2 myofibrils in parallel, and you can see that this is in passive conditions, this is in activated conditions. There will be a slight shearing, but not big enough, and they're mostly connected.
I see. Okay, so I guess the picture in my head is that I do agree that sarcomeres in series with each other might not receive much information about, a sarcomere 10 sarcomeres down the line is stretched more than I am, and so we need to equilibrate or something. But I do think that if there's this stretchy material between neighboring myofibrils, then there's another uniformity that could be enforced, which is one myofibril, or actually one sarcomere in a myofibril might be induced. It can't differ too much from what its neighbors are doing. If they all shorten together, then they're all happy in that area. But if one sarcomere shortens, or let's say lengthens, because we're interested in the instability. So let's say it lengthens more than its neighbors, then I would expect that the extracellular matrix would tend to be pulling it against that. And again, with the cycling of the cross bridges, that sort of would be expected to give you that downward slope that you see. So that's a parallel uniformity, which could look like it fixes a series non-uniformity, but that it all kind of goes together.
That is a sound explanation. I like that. So maybe we can do some modeling study on this to prove that that's actually the case.
Yeah, I think it wouldn't be hard to. I mean, the way I would do it is I would do it with, say, two neighboring myofibrils with some stretchy material, and just demonstrate how 2 or 3 of these would have that effect, rather than try to model entire fibers and multiple fibers and things like that. But it would just be a conceptual indication of a possible explanation.
Mm-hmm. Great, thanks for the suggestion. That's great.
Since I am running out of time, so Rob, if you have a super quick question, please go ahead.
Quick question. The resolution, you have good resolution of length for the sarcomeres, I'm not sure what resolution you have for measuring the force. If you've got about 20 sarcomeres in series and some of them are wandering, and perhaps wandering from producing active force to their force coming from passive mechanisms, do you see a trembling of the force there? And so you had instability decreasing over the 600 milliseconds. Would you see a sort of a trembling of the force with the instability to begin with that then becomes stable? Or if you don't see a trembling of the force, are you simply wandering for a single sarcomere producing active force, but then becoming unstable, but then the force is still the same because the length changes, but your force is still maintained by passive mechanisms? My guess is you simply won't have the resolution in the force to see any trembling in the force measurement, but have you looked? Or
we don't have a different type of muscle force transducer. So what you are seeing at the moment here, that is a force trace that we collected. We don't see that. Maybe there is some vibration, but we cannot separate it out from the ambient noise. Yeah, so we need a higher sensitivity force transducer.
Yeah, that's a very smooth force trace. It's yeah. I would expect a noisier one.
I'm not sure. I think, did I filter it? I probably filtered it a little bit. Yeah.
Okay, on that note, I would like to thank Eng Kuan again for sharing his great work.
Thank you for the chance to present my work today. Someone, thanks.
Thanks,
yes.
Eng Kuan.
Thanks, Eng Kuan.
Thank
Thanks
you.
So
Thanks.
actually, let me explain about the next seminar. Yeah.
Sorry.
we will be hearing from Dr. Delight Polay, and the title is Locomotion as an Optimization Process. So thanks for coming today again, and I will see you all next week.